Hey microbiology students, we're starting a new lecture series this week on pathogenic bacteria. I call this part 2, and in this lecture series we talk about the bacterial pathogens of the various body systems. Now I thought long and hard about how to deliver these lecture notes to you through distance learning because usually, and actually over the years, I have eliminated more and more the PowerPoint notes, and I do more and more illustrating on the whiteboard and that sort of thing. However, in this distance learning format, I decided that I really needed to utilize PowerPoint as best I could in order to convey this information to you. And I also decided to provide you with all of the lecture notes that you're about to see me present in these videos. And so hopefully you do watch the videos. I encourage you to watch my videos. However, I encourage you to have the lecture notes, the PowerPoint notes in front of you as we go through them. Maybe highlight key terms. I will write some occasional notes on the slide myself, so you should write those on your slides as well. In addition, I might mention some things that aren't on the PowerPoint slides, so you're going to want to pay close attention to those extra details. And as always, please follow along with your learning objectives for each lecture that you watch so that you make sure you catch all the important information that will be on your next exam. The next exam is very case study heavy. Most of my microbiology students really enjoy this part of the semester because you are, of course, pre-allied health majors going into medical professions. And so most of you have an interest in infectious disease and find this to be very fascinating. As well, I present you with cases where you're given patient histories and you have to solve what mystery disease these patients have and understand things like the treatment of the disease, prevention of the disease, is it vaccine preventable, those sorts of things. Certainly anything that's presented on the lecture slides, as well as anything in addition that I mentioned during the lecture video, could show up on the exam. I may also apply principles that we're learning about in this lecture series to a completely different bacterial disease on the lecture exam and ask you to utilize some critical thinking skills in order to make some connections that's based on information that you should know from our pathogenic bacteria unit. Please note that we are not going to be discussing every single bacterial pathogen known to man, okay, in this new lecture series. We would be here a really long time, wouldn't we? If we did every single bacterial pathogen known to man, it'll be quite a long list once we get through all these lecture videos, but these are the ones. So the ones that I've selected for our video series are the ones I think are the most important to know as a future health professional, okay? So the way I've structured these videos, what will happen is we will take on essentially one body system at a time, do a really general overview of the anatomy of that body system, really basic, okay? Talk about some of the key features, especially the immune defenses involved with that body system. And then we will go through some of the selected diseases that I've chosen to talk to you about. Okay? So let's get started. You can tell here from the screen that we are going to be addressing in this lecture video bacterial pathogens of the gastrointestinal system. So let's start by an overview of the digestive tract and what do we mean by that? Well, it actually starts in the oral cavity. Remember, though, that your mouth is home to many diverse microflora. It is a major portal of entry because that is obviously, the nasal cavity, the oral cavity. That is, how we breathe. It's also where we eat and where we drink. And so it's a major portal of entry for pathogens. We'll start there. We'll talk about some dental, particular dental pathogen. Venturing then from the mouth down into the stomach, we've talked before that the stomach acid acts as a chemical defense, part of the first line of defense of the immune system. And for the most part, it kills just about all microbes with some exceptions. Next up would be the small intestine. Here we get lots of enzymatic secretions coming from the pancreas. It kills most of your microbes. Peyer's patches, this is a collection of leukocytes, and they're there to help prevent any overgrowth of bacteria in the small intestine. There really shouldn't be any bacteria in the small intestine. The large intestine, like the mouth, is also a location in the body where we have a lot of our microflora, large quantities of E. coli and a collection of other types of microorganisms. We've talked before about why that's really an important thing. Not only do they help you with the digestive process, they produce essential vitamins like vitamin K. There's a principle called competitive inhibition where we've talked about it before. It's like the garden. Remember, so if you have a good garden of gut microbes growing in your large intestine, it keeps the weeds out. It prevents the pathogenic bacteria from growing in your gut. So eating foods like fermented foods that are rich in bacteria, for example, like kombucha or yogurt or fermented cabbage, all of those have the good bacteria. We call them the probiotic bacteria that colonize your large intestine. And you know what? You want them to be there. You want them to be present because they are the beautiful flowers in the garden of your gut microbiome, and they're going to keep the weeds out. Okay, so let's start in the mouth. Here's Streptococcus mutans. Streptococcus mutans is in the genus Streptococcus, so although not always, the genus name sometimes refers to the morphology of the bacteria. So this is the arrangement of streptococci. So those circle-shaped, what we call cocci-shaped bacteria in a chain. And we actually, in the beginning of the semester, did a mouth bacteria lab where you gram stained some of the bacteria from your mouth. And then remember, I took a swab out of my mouth and we looked at it under the dark field microscope, and we saw my happy little creatures from my mouth swimming around. Okay, so we have a host form of lots of good bacteria in the mouth, but Streptococcus mutans, you know what? It's an opportunistic pathogen. What that means is, if you're brushing your teeth regularly and like twice a day, okay, flossing once a day, those sorts of activities, and not eating a lot of sugary food, that will reduce the amount of Streptococcus mutans in your mouth. Do you remember part of that mouth lab that some of you had your spit tested to see if you have strep mutans in your spit? And do you remember how we figured that out? So it grew in this test tube, and if the test tube turned yellow with cracks in the agar, that indicated fermentation. And so, of course, strep mutans, if you give it the opportunity, again, opportunistic pathogen, if you give it an opportunity, like lots of sugary food and no brushing of the teeth, oh, it's going to love that. It'll just love it, and love it so much, it's going to ferment the sugars. And we know from studying fermentation that one of the products of fermentation is in fact lactic acid. So lactic acid gets produced and that degrades the tooth in the animal. And there you go. We get tooth decay, also known as dental caries. So going to see your dental hygienist, recommendation is about every 6 months. go see your dental hygienist. They do a deep clean of your teeth. They remove plaque because of the biofilms of bacteria that accumulate on our teeth. Even if we are practicing good dental hygiene, certainly you can get large biofilms forming. And so that's the purpose of our dental hygienists that get in there. They remove excess plaque, or known as tartar. Here we could see in these diagrams pictures of people who have severe tooth decay, and it's just showing you, of course, eating away at the enamel, deep down even into the root of the gums. It can get really, really severe where people need root canals and more than just little fillings that can, fill in these holes that form in the teeth, but you can get really severe infections that can, because the bacteria can get deep into your the root of the tooth and into the blood supply and all sorts of things. So you don't want bacteria growing in your blood. Let's move now into the stomach. Helicobacter pylori, gram-negative Vibrio. Remember, Vibrio means sort of like a curved rod shape. So these bacteria colonize the stomach. Now, how do they do that? Because the stomach is so acidic, right? Well, they have certain adaptations that have allowed them to colonize the stomach. They tolerate the low pH by secreting a buffer around themselves, and they do that using a specialized enzyme we call urease. Okay, so some pathology associated with H. pylori is inflammation of the stomach that can lead to ulceration. Although many people, here we see greater than 80% are asymptomatic, or not showing symptoms of the infection. And some people develop the peptic ulcer disease or the stomach ulcers. And a very small percentage of people actually develop stomach cancer. Transmitted fecal to oral route, okay, is what we suspect from human to human. And roughly 50% of the world is actually infected with H. pylori. H. pylori is pretty famous because of the scientist Barry Marshall, who won the Nobel Prize for discovering that H. pylori causes peptic ulcer disease. But he did something very outlandish where he actually drank a culture of H. pylori. How horrifying is that? Think about those broth cultures that we grew in the lab at the beginning of the term. Imagine drinking that. That was highly not recommended. Okay, glad you didn't do that. Well, he did that to prove that he could get ulcers from the bacteria. You see, people thought that ulcers were caused by stress or spicy food and not caused by a bacteria. And Barry Marshall, he determined that it was in fact caused by this little bacteria. Here's a picture of what can happen in the stomach when we get invasion of the stomach mucosa by H. pylori. And remember, I have my collection of giant microbes, my little plush, stuffy toys of the bacteria that I like to talk about in my class. And now I just embed them into my PowerPoint slides so we can see the cuteness of the plush version of the pretty horrifying microbes that we'll be discussing. So this is the plush form. This is the giant microbe form of H. pylori. You could see the hair. Remember what that hair represents? Flagella, right? So it's a flagellated microbe. It can, it can swim around and escape the immune cells. This is where I began my obsession with giant microbes, was H. pylori. So taking you back years and years ago, gosh, how long has it been now? Oh gosh, it's been almost 13 years since I was in graduate school at UC Davis, researching H. pylori as a microbiology grad student, deciding what I was going to do with my life. Falling in love with teaching, by the way, is when that all played out. I decided research, not so much. Didn't enjoy being in the lab all by myself. Of course, now I'm teaching distance learning, and I spend most of my time all by myself, talking to myself. and creating these videos for you, which is feels weird most of the time. But anyways, so I fell in love with teaching, and um, I knew that I wanted to educate people, especially in microbiology. And somebody gave me a giant microbe of H. pylori at that time, since that was what our lab researched. And that started my obsession, where I wanted to get every little creature I could get my hands on, mostly because my students would say to me, "Oh, microbiology, that's so abstract, and you can't hold the microbes in the palm of your hand like you can when you're studying anatomy." And I said, "Oh, well, yeah, but these giant microbes, that we can hold in the palm of our hand. We can look at the structures of the bacterial cells, and we can learn some things. They're actually educational. Besides being cute, these giant microbes are also very educational." So this, of course, is a flagellated cell. Okay, so now let's move into the intestines, pathogens of the intestines. I don't know how many of you have experienced food poisoning. Pretty terrible, okay? I've experienced food poisoning several times in my life. And boy, is it intense, okay? Where you realize that you ate something that was contaminated with some bacteria, or was contaminated with bacterial toxins. So we'll talk about that next here, which is that if you have inflammation in the intestines caused by ingesting food or water contaminated with bacteria, it may be the bacteria themselves that are getting into your intestines and causing the pathology, but it may be that the food that you consumed was just contaminated with enterotoxin. So in last week's video, we talked about the different types of toxins produced by bacteria. It's an example of a virulence trait. Recall, there are exotoxins, which are secreted by both gram-positive and gram-negative bacteria. And then there are endotoxin, which basically is the cell wall of gram-negative bacteria. Okay, so enterotoxins are secreted by many different strains of bacterial pathogens. However, the two that you see pictured on this slide are the two culprits for enterotoxin food poisoning. So what happens is you ingest food that is contaminated with enterotoxin. And so usually this is food that you've prepared by hand, because both of the bacteria that you see here, this is Staph aureus in purple, looking like purple grapes, and Bacillus cereus here in the bottom. This is the bacillus-shaped bacteria. So both of these actually colonize people's skin pretty regularly. And so what happens is, usually food that you prepare by hand, or if somebody has been eating that food, and sort of like in guacamole, right? So if you get guacamole for a party, and this is why you're not supposed to double dip, right? If you take a chip and you dip it into the guacamole, you shouldn't re-dip, because the chip now has your mouth bacteria on it, or skin bacteria on it. And then what can happen is, let's say that food is left out in a warm environment. So bacteria love warm environments. They love warm environments. This is how it speeds up their growth. So food, like at a picnic, picnics are oftentimes places where people get enterotoxin food poisoning later, because there's lots of people, like a buffet or something like this, picnic, where everybody is dipping into the same container, or maybe double dipping with their mouth and contaminating it with their mouth bacteria. And then you have it, left out in the sun for a long, many hours or something like that. What happens is the staph bacteria, or the bacillus bacteria, they just happily grow in that food. I'm thinking like potato salad or guacamole or something along those lines, some sort of food, and they just grow out of control, basically, and they just secrete large quantities of enterotoxin. So they're just cranking this toxin out. And so the food becomes contaminated with lots and lots of enterotoxin. Interestingly enough, that doesn't affect the taste of the food. Okay, so you're usually, not always, but usually you can't really tell in the process of eating the food if it is contaminated with enterotoxin. Um, Enterotoxin is oftentimes heat stable. So even if the bacterial cells, maybe if the bacterial cells get cooked, in the sun, you have it out in the sun, and the cells might start dying off because they're getting intense heat, the enterotoxin is heat stable. So it'll stay in the food. So once you eat that food, you're likely not consuming very much bacteria, if any bacteria at all. You're consuming large quantities of enterotoxin. And that toxin is going to be fast acting on your intestinal cells. It's going to be fast and it's going to be bad. Okay. So we're going to see vomiting and diarrhea and cramping, and you're going to feel really, really bad. But the good news is when it's enterotoxin food poisoning, it's rapid recovery. Okay. So here we have on the slide a very short incubation period. Remember, that terminology means from the time you come in contact with the bacteria or the disease agent. Here we're talking about a toxin. So you're actually ingesting the toxin, not the bacteria. Then we have a really short time until you show symptoms, until you start getting the cramping in your intestines and you start, wow, I've got to vomit. Wow, it's going to come out both ends. Wow, right? By the way, this is the poop lecture, so get nice and comfortable. Hopefully you're not eating lunch right now while you're watching this show. This is the poop lecture. We go into way more detail than you probably ever wanted to know about poop. Okay. All right, 1 to 3 hours, you have your symptoms. Rapid recovery, usually lasting about 24 hours, even though people feel terrible. And the times that I've had enterotoxin food poisoning, once I can remember fairly recent. The last time was actually at my daughter's first birthday party. We went to Chuck E. Cheese, and it was the pizza. Must have been handled and left out, and there was enterotoxin poisoning going on there. And just as soon as I got home, it had just been an hour or so, that's when all these symptoms just, wow, started in me. Don't resist the vomiting, because you need to get that poison out. Okay, that's one of the lessons that I've learned from enterotoxin poisoning, is that when you have that urge to vomit as a result of this poisoning, please do so. Do not resist, because that is the body's way to get the poison out. You have large quantities of poison. And what this poison does is it attaches to your intestinal cells, and it makes those intestinal cells leak fluid. Okay. And so what you need to do is, the vomiting and the diarrhea does flush out the toxin. In the majority of the time, it's not something that's going to require antibiotics. It's not going to require hospitalization. It'll be, your body will clear the infection within 24 hours. Okay, contrast that with bacterial enteritis, also known as gastroenteritis. So what's similar? This is also food poisoning. It can be. And it also has the same sorts of symptoms associated with enterotoxin poisoning. So you're still going to get the nausea and the vomiting and the cramping and the diarrhea and all of this. Okay. The difference, notice in the definition, we're telling you here that enteritis is a bacterial infection of the intestines. It's caused by the bacteria. Now, they may or may not also be secreting toxins, but we do differentiate this from enterotoxin food poisoning in that it's the bacteria that are causing the damage. So they're colonizing. Eventually, they do produce many types of toxins, though, that cause damage. Notice some other differentiating criteria. It takes longer for the symptoms to appear. That's because the bacteria are usually ingested either in food and water in small quantities, like maybe just a few cells. And then the cells have to get past your immune defense, and then they have to get to their target organ, which is the intestines. and then they need to grow. And remember, bacterial growth, although can be exponential, it usually is about a 24 to 48 hour process for the bacterial numbers to get really high. And they'll get really high, right? They'll get into the billions, fairly quickly within a day or two. So this is highly variable. There's several different pathogens that cause gastroenteritis, so we're going to talk about them next. So it's generally longer symptoms, it's generally more severe, and it lasts longer, often associated with a fever, often associated with very severe diarrhea that we call dysentery. Using that word dysentery is different than using the word diarrhea. So dysentery usually refers to very severe diarrhea, and it can be accompanied by blood or mucus in the stools. So let's meet the pathogens that cause gastroenteritis. All right, first up, Salmonella. Okay, so this is Salmonella enteritis or Typhimurium. So there's actually various subtypes of bacteria within the genus Salmonella. You can see here it's a gram-negative rod. This is the giant microbe version of it, so it's flagellated. So they replicate inside of the cells of the small and large intestines. This causes a huge inflammatory response where we get diarrhea. They can enter the blood, bacteria can enter the blood, and then a systemic infection means that they can travel throughout the body. So you may know that Salmonella has a natural reservoir in chicken, in the guts of chicken. So this is why you want to be very careful when you're handling chicken in the kitchen, raw chicken. So being careful what you touch when you have your chicken hands. I call it chicken hands, okay? So when I'm cutting up raw chicken in the kitchen, I'm very careful about what I touch, including faucets and doorknobs and knives and all of those things that I know that I've touched. I very carefully go back and disinfect later because of the possibility of having salmonella on my hands and then contaminating an object, a fomite, with the salmonella. This type of salmonella is not as severe as its cousin. We'll meet its cousin in a minute here. So we say transmission is fecal to oral. So that means that you either eat something that's contaminated with salmonella, like undercooked chicken. This is also why meat thermometers are really essential part of your kitchen, because like when you cook Thanksgiving dinner, you probably know what, that you're supposed to get the turkey to a particular temperature using a meat thermometer. Because if you don't cook it to the right temperature, then salmonella bacteria could be in the undercooked chicken. I tend to always overcook my chicken. It makes my husband crazy. He says, "You've overcooked the chicken again." I know, but I want to make sure I kill all the salmonella. So it's hard for me to undercook chicken, knowing what I know. Anyway, so contaminated chicken, eggs, also reptiles. You may not realize this, but snakes and lizards, salmonella is a normal part of their skin flora. So if you have these types of pets, then you may be picking up the bacteria. And like I was saying, not contaminating objects in your kitchen. Remember, objects, surfaces, we call that fomite transmission. So fecal to oral means somehow either you're eating something that's contaminated or you touch your mouth after touching an object that was contaminated with the bacteria. So it's a type of indirect transmission. Salmonella typhi, this is what I meant by this is the cousin of Salmonella enteritis and Salmonella typhimurium. Bet you don't want to meet this one. Notice in the giant microbe, they made this one red. So this one's white, this one's red, meaning it's a more severe, like alarm, red usually means like alarm, severe. Okay, so what does this guy do? Well, he grows in macrophages, great. So he's got that going for him. That protects him, so he's able to grow inside of the macrophage and not be digested by the macrophage. He can spread to other organs, he can spread to the blood, it can go systemic. Very high fever associated with typhoid fever, essentially, that's where it gets its name. Eventually perforating the intestinal wall. Same sort of thing. It's going to be fecal to oral transmission. 20% mortality if untreated. Okay, with some possibility of chronic carriers. The very famous one that I mentioned last week in the lecture videos was Typhoid Mary. She was the cook in the early 1900s in New York City who was associated with many dozens of deaths as a result of her passing the salmonella in her food. She likely wasn't washing her hands effectively after using the restroom. Okay, Shigella, cause of shigellosis, another gram-negative rod. Here we could see in the giant microbe that it's got fimbriae, right? The little sticky hairs. So it resides in human guts. Don't need a lot of it. That's what low minimum dose means. You don't need a lot of it in order to get this infection. Does a lot of the same things that salmonella does, including invading the cells of the large intestine. However, it doesn't typically go into the blood. Okay, you can get really severe symptoms, including the dysentery. Here's a virulence trait you should take note of. It's called the Shigella toxin. It's a neurotoxin associated with convulsions, coma, and death. E. coli. So E. coli and Shigella are actually pretty similar. Some microbiologists think that we should actually reclassify Shigella as just a subtype of E. coli because they're so very similar. So E. coli, there's lots of strains of E. coli. And we've talked about it before because it's a part of your normal microflora. And it's usually, an example of a mutualist, meaning it actually provides you with benefits like digesting your food and producing certain vitamins. There's only a few strains that are pathogenic. It's possible to just get mild gastroenteritis. We call that traveler's diarrhea. That means that when you travel to other parts of the world, a lot of times people are somewhat immune to the strains of bacteria that are endemic or common in certain areas. So you move into that area or you visit that area and maybe you get sick, but other people don't usually get sick because they're sort of immune to those bacteria. This is really common in Mexico, actually. It's one of the reasons why, when we travel to Mexico and eat the food there, sometimes those of us who aren't accustomed to that type of E. coli, we might get some mild gastroenteritis, but the local people would not. Of course, there's a dangerous version of E. coli you should be aware of. It's called E. coli 0157H7. This is the highly virulent strain. This is the one that makes headlines every once in a while, showing up in certain fast food chains. It produces a Shigella-like toxin, so again, it's really closely related genetically to the Shigella bacteria. The really dangerous disease that can result is called hemolytic uremic syndrome, and what happens is the bacteria invade the kidneys and you get massive kidney failure because of all the inflammation. Usually these people will be, they will have bloody stools. So you may recall seeing E. coli under the microscope earlier this semester. That's because E. coli, like I said, most of the time it's non-pathogenic. It can act as an opportunistic pathogen. And then there are virulent strains, and what differentiates a virulent strain from a non-virulent strain, it's usually a genetic reason. There's these virulence factors. So that means that the bacteria that are just your garden variety E. coli that live in your large intestine, you know what, they don't have the Shigella toxin. Okay, so that's why they're not causing disease in you. So it's gram-negative, okay, with fimbriae, with flagella, and they look sort of like little Tic Tacs under the microscope, little pink Tic Tacs. Let's see more about the hemorrhagic E. coli, like the 0157H7 strain. So this is normally found in cattle guts. So it's a normal flora for the cattle, and so it transmits to us usually in contaminated beef. Okay, so that might be beef that's contaminated with feces from the cattle. Again, those of you who like to eat your hamburgers rare, just a word of caution there is that if you do like your meat rare, you are setting yourself up for potential infection with infectious diseases. Most of the time, your immune system will probably handle it for you without you even knowing it. But if you're immunocompromised, like if you're pregnant, if you have some other condition going on that has weakened your immune system, you might want to consider not eating your beef rare. Sometimes in the news we learn about E. coli poisoning on vegetables or fruit. That has to be from feces contaminated, usually water. So there's been recalls here in California a few years ago, spinach. So spinach was contaminated with E. coli. And if you're not properly washing your vegetables before you eat them, then you could be potentially exposing yourself to pathogenic E. coli. Okay, so here's some more of the virulence factors of E. coli. Okay, next, cholera. Cholera is caused by Vibrio cholerae, gram-negative Vibrio, flagellated, lives in water, transmits from contaminated water. This is usually where water sanitation is not present. Cholera definitely has a historical context because it's been around a really long time in the history of humanity, and there's lots of historical cholera epidemics, especially in Europe, like in the 1800s and prior to that as well, where lots and lots of people would die. So the virulence of cholera is due to the toxin. So it also produces an exotoxin that binds to the stomach, or sorry, not the stomach, the intestines, and causes water loss. Okay, and that the main concern with cholera is the severe dehydration that results. And there's also a very characteristic type of diarrhea that's produced by cholera, and it's watery. Okay, it's the watery stool. Here, it's a lot, like 15 liters per day. Back in the old days, what they would do during cholera epidemics is they would actually lay their patients down on just a cot with a bucket at the on the rear end, so they would just be spewing out all of this cholera infected poop all day long, 15 liters, and it's extremely watery. We oftentimes refer to it as the rice water stool. So that imagery is if you've ever washed rice before you cook it and you get sort of that sort of cloudy water, that's what the diarrhea looks like for cholera. So the major concern is death caused by rapid dehydration, leading to organ failure and death. Very high mortality, 50% if you're untreated. There is a preventative vaccine. So there's an oral cholera vaccine that can be given, although there are still epidemics of cholera every once in a while in the news because not all countries vaccinate their populations. Okay, so that brings us to the end of this lecture video on pathogens that infect the gastrointestinal tract.